Hi everyone, my name is Sarah, and today I want to explain a common molecular genetics technique used in research called reporter genes. Specifically, I'd like to discuss the differences between transcriptional and translational reporter genes. To get started, I first need to explain the central dogma of molecular biology. This is the concept that DNA is transcribed into RNA, which is then translated into protein. DNA carries genetic code, but proteins are the working units that carry out important functions inside cells. So what are reporters? Reporters are genes that are fused to genes of interest, abbreviated GOI, to monitor their activity at the transcriptional or translational level. Transcriptional reporter genes are fused to the promoter of the gene of interest, which is depicted in Figure A below. Translational reporter genes, on the other hand, are fused to the gene itself, seen in Figure B. The reporter could be fused to just a segment of the gene or the entire gene. Again, transcriptional reporters are fused to the promoters of genes, which provides information on where the gene is transcribed, but not where the translated protein ends up. Here you see an embryo that has a transcriptional reporter. Translational reporters that are fused to the entire gene of interest provide information on where the translated proteins are located, and not necessarily where the RNA was transcribed. This slide lists some of the most common reporters used in research. Both green fluorescent protein, or GFP, and red fluorescent protein, or RFP, are genes that encode fluorescent proteins. Luciferase, beta-galactosidase, encoded by the gene LAC-Z, and beta-glucuronidase, encoded by the gene GUS, are enzymes that are involved in a chemical reaction to produce light or color. GFP is probably the most commonly used reporter and was originally isolated from the crystal jellyfish on the west coast of North America. Interestingly, in 2008, 3 scientists won the Nobel Prize in Chemistry for their discovery of GFP. GFP encodes a 26.9 kilodalton beta-barrel protein that fluoresces bright green under UV light. To check your understanding, here are a few sample questions that you should be able to answer. Feel free to pause the video if you'd like more than a few seconds to think about each question. First, what kind of reporter, transcriptional or translational, would you use to determine the expression pattern of a gene of interest? You could argue that either could be used depending on whether you want to visualize where the RNA is transcribed or where the protein is expressed. The next question is, what kind of reporter would you use to determine the subcellular location of a protein of interest? The correct answer would be translational, because the single fused protein of interest in GFP will enter the compartment that the protein of interest localization signal tells it to go. Finally, what kind of reporter would you use to test protein mobility between cells? The correct answer would be both, because we have to know both where the gene of interest was transcribed and where the translated product ends up to determine mobility. Thank you for watching.